Hello, welcome to Biology, the science study of life. Molecular systematics is illuminating prokaryotic phylogeny. Until the late 20th century, systematists based prokaryotic taxonomy on phenotypic criteria such as shape, motility, nutritional mode, and response to gram staining. These criteria are still valuable in certain contexts, such as the rapid identification of pathogenic bacteria cultured from a patient's blood. But when it comes to prokaryotic phylogeny, comparing these characteristics does not reveal a clear history. Applying molecular systematics to the investigating of prokaryotic phylogeny, however, has produced dramatic results. Lessons from molecular systematics. Microbiologists began comparing the sequences of prokaryotic genes in the 1970s using small subunit ribosomal RNA as surRNA as a marker for evolutionary relationships. Carwes and his colleagues concluded that many prokaryotes, once classified as bacteria, are actually more closely related to eukaryotes and belong to a domain of their own, Archaea. Microbiologists have since analyzed larger amounts of genetic data, in some cases entire genomes, and have concluded that a few traditional taxonomic groups such as cyanobacteria are in fact monophyletic. However, other groups such as gram-negative bacteria are scattered throughout several lineages, shows a tentative phylogeny of some of a major taxa of prokaryotes based on molecular systematics. Prokaryotic phylogeny is still a work in progress, but two important lessons have already emerged. One is that the genetic diversity of prokaryotes is immense. When researchers began to sequence the genes of prokaryotes, they could investigate only those species that could be cultured in the laboratory, a small minority of all prokaryotic species. Methods pioneered by Norman Bass of the University of Colorado in the 1980s now allow researchers to sample genetic material directly from the environment. Every year, this genetic prospecting adds major new branches to the tree of life. Some researchers suggest that certain branches represent entire new kingdoms, while only 4,500 prokaryotic species have been fully characterized. A single handful of soil could contain 10,000 prokaryotic species. According to some estimates, you can see why taking full stock of this diversity will require many years of research. Another important reason is the apparent significance of horizontal gene transfer in the evolution of prokaryotes. Over hundreds of millions of years, prokaryotes have acquired genes from distinctly related species, and they continue to do so today. As a result, significant portions of the genomes of many prokaryotes are actually mosaics of genes imported from other species. Note again in this figure a key inferences based on molecular systematics. The very early divergence of prokaryotes into two main lineages, bacteria and Archaea. Bacteria. Let's begin. All start with bacteria. This domain, bacteria include the vast majority of prokaryotes that most people are aware of, from the pathogenic species that cause strep throat and other diseases in the beneficial species used to make Swiss cheese. Every major mode of nutrition and metabolism is represented among bacteria, and even a small taxonomic group of bacteria may contain species exhibiting many different nutritional modes. On the following also is two pages we can closer look at several major groups of bacteria. Ehrlichia share certain traits with bacteria and other traits with eukaryotes. However, Ehrlichia also have many unique characteristics, as we would expect for a taxon that has followed a separate evolutionary path for so long. The first prokaryotes that were classified in domain Ehrlichia are species that live in environments so extreme that few other organisms can survive there. Such organisms are known as extremophils, meaning lovers of extreme conditions, from the Greek philos, lover. Extremophils include extreme thermophils, extreme halophils, and methanogens. Extreme thermophils from the Greek thermo, hot, thrive in very hot environments. For example, Ehrlichia in the genes sulfobas love or live in sulfur-rich volcanic springs at temperature up to 90 Celsius degree. Also, another type called scientific name Pherolaps fomeri, an extreme thermophile